Hello students, welcome back to biology class. In the previous classes, we discussed about the male and the female reproductive systems. We also discussed about what is gametogenesis and also about spermatogenesis. Right? Now today, let's discuss about oogenesis. Now you all know the meaning of genesis. Genesis means synthesis. O stands for ovum. So how can we define oogenesis as? I hope you all can define it. It can be defined as the synthesis of ovum. Now where does the synthesis take place? It takes place within the ovary. Exactly where inside the ovary? Inside the follicles of the ovary. So the synthesis of ovum within the follicles of the ovary is called as oogenesis. Or in other words, we can define it as the process of formation of mature female gamete. Mature female gamete. What is a female gamete? It is the ovum. So that is the definition of oogenesis. Now, we have to study in detail, or let's see what are the steps involved in the process of oogenesis. Now when we discussed about spermatogenesis, we saw that spermatogenesis begins at puberty, means when a boy reaches the age of 13 or 14. Whereas oogenesis begins during embryonic development. Embryonic development means when a female baby is being formed inside the mother's womb, that time itself oogenesis begins. Clear? So oogenesis begins during embryonic development. Now, millions of oogonia. When we discussed spermatogenesis, we saw that spermatogonia are the male germ mother cells. Here, oogonia are the female germ mother cells or the gamete mother cells. These oogonia are formed in each fetal ovary. What does it mean? Inside the ovary of the fetus, when the fetus is developing inside the mother's womb, that time itself millions of gamete mother cells or oogonia will be formed. Now this oogonia within the womb itself, that is within the fetus itself, it start division and it enter into prophase 1 of meiotic division and get temporarily arrested at that stage called primary oocyte. What does it mean? So as I've told you, the oogenesis process of synthesis of ovum begins as soon as the embryo is formed in the mother's womb. Female embryo is formed in the mother's womb, and when in the mother's womb itself, the fetal ovary will be having millions of oogonia, and this oogonia start division. Which division? Meiotic division. There are two stages in meiosis, that is meiosis 1 and meiosis 2. So in under meiosis again there are 4 different phases. Which are they? Prophase 1, metaphase 1, anaphase 1 and telophase 1. So the prophase 1 of meiotic division begins when the within the fetal ovary itself. So the prophase 1 of meiotic division takes place in the oogonia, and after prophase 1 it gets temporarily arrested there. And now what is formed? The oogonia after prophase 1, that is called as the primary oocyte. So when a baby is born, when a female baby is born, the baby will be having number of primary oocyte. Okay, hope it's clear. Now let's see the schematic representation of oogenesis. Just as we have studied in spermatogenesis, here also in oogenesis also the division will be the same. That is, in the beginning the oogonia undergoes number of mitotic division, resulting in the formation of number of oogonia. This takes place within the fetal ovary. After that, the next is what? The beginning of the first meiotic division. So the first meiotic division takes place within the fetal ovary itself, right? when the baby is inside the mother's womb, that time itself, the prophase 1 of meiosis 1 will be completed, resulting in the formation of primary oocyte. So when a baby is born, there will be millions of, not millions, there will be millions of oogonia or the primary oocyte, okay, within the baby's ovary. Now this primary oocyte undergoes or completes the first meiotic division, resulting in the formation of two cells. But here the division, first meiotic division as well as the second meiotic division is unequal. Means after the division, two unequal sized cells will be formed. The cells formed will not be of the same size. One will be a smaller cell and the other one will be a bigger cell. So the primary oocyte, when it completes the first meiotic division, it results in the formation of a smaller polar body and a large secondary oocyte. So here you can see. So this is the primary oocyte. So when a baby is born, during birth, the baby will be having number of primary oocyte. Now this primary oocyte, by the time when the child attains puberty, the girl child attains puberty, the primary oocyte undergoes or completes the first meiotic division, which is unequal, resulting in the formation of two cells. First one, a small polar body, which is called as the first polar body, and a large cell, which is called as the secondary oocyte. Now this secondary oocyte begins, or the second meiotic division begins in the secondary oocyte, but this takes place only this secondary oocyte undergoes meiotic division, begins meiotic division and gets arrested in the metaphase 2 stage, okay, in the metaphase 2 stage, and will be released out. Now the division of the secondary oocyte or the meiosis 2 will be completed only after fertilization or only after the entry of the sperm into the ovum. Then only the secondary oocyte completes its meiotic division. So this, it is actually the sperm entering into the secondary oocyte. Even though we call that as an ovum, it is not the ovum which is completely formed, it is the secondary oocyte. So the secondary oocyte completes its meiotic division, resulting in the formation of a functional ovum and again a second polar body. So if we see only the divisions, it is somewhat similar to that of spermatogenesis. Only one difference is there. Here it is unequal. Meiosis is unequal. There the meiosis is equal. So once again, so what happens is, first oogonia will be formed within the fetal ovary. Millions of oogonia will be formed, and this oogonia undergoes first meiotic division, that is only prophase 1 of first meiotic division, resulting in the formation of primary oocyte. This primary oocyte later completes first meiotic division, which is unequal, resulting in the formation of a small cell with a, which is called as the first polar body, and a large cell called the secondary oocyte. So primary oocyte will be having 46 chromosomes and the secondary oocyte will be having only 23 chromosomes. This secondary oocyte, when it completes its meiotic division, that is when it completes meiosis 2, which is again unequal, it results in the formation of a second polar body which is small and a large cell which is called as the ovum. So here also we can divide this into three different phases. That is, this is a phase of multiplication, then this is a phase of growth, and this is a phase of maturation. So this is a schematic representation. You have to note all these. Okay. So that is division of these oogonial cells takes place during the fetal life itself. That is within the fetal ovary. Now when the baby is born, the baby will be having primary oocytes. Now after birth, when the baby attains puberty, then slowly the first meiotic division will be completed, resulting in the formation of a first polar body and a secondary oocyte. And now this undergoes second meiotic division, resulting in the formation of a second polar body and ovum. This takes place only during the adult reproductive life. That means after fertilization. Okay. Now let's see a little more in detail. So as we have seen, in the fetal ovary there will be number of oogonia, and this oogonia undergoes prophase 1 of meiosis 1, resulting in the formation of number of primary oocytes. So this is a section. In this picture, you can see the section of an ovary. Now here you can see these are the primary oocytes. Okay. Now this primary oocyte slowly gets surrounded by a layer of granulosa cells and is now called the primary follicle. So a primary oocyte surrounded by number of granulosa cells is called as a primary follicle. Hope it's clear. So what is a primary follicle? A primary oocyte surrounded by number of granulosa cells. This primary oocyte now goes on around the primary oocyte, number of granulosa cells will be formed. So at puberty, there will be about 60,000 to 80,000 primary follicles in each ovary. So a girl child, when she attains puberty, she has 2 ovaries, and in each ovary there will be about 60,000 to 80,000 primary follicles. So when the baby is born, millions of primary oocyte or oogonia will be found. But by the time she attains maturity, only 60,000 to 80,000 primary follicles will be left in each ovary. The remaining ones get degenerated. Hope it's clear. Right. Now the primary follicle, the oocyte, the primary follicles, that is, these are the primary follicles, get surrounded by more layers of granulosa cells to form secondary follicle. So a primary follicle transforms into a secondary follicle by formation of number of granulosa cells around the primary oocyte. Now these granulosa cells now differentiate into a covering, that is external theca and an internal theca. Theca means covering. So it forms a cover around the primary oocyte. Now the secondary follicle slowly transforms into a tertiary follicle. So by the time a primary follicle gets transformed into a tertiary follicle, a cavity will be developed inside, and the primary oocyte will be pushed towards one side, and it will be surrounded by 2 to 3 layers of granulosa cells, which we call as the theca externa and theca interna. So theca externa and theca interna is formed by granulosa cells. Hope it's clear, right? And the cavity which is developed within the follicle, tertiary follicle, is called as the antrum. So what is antrum? It is nothing but the cavity which is formed within the tertiary follicle. Hope it's clear. So a primary follicle slowly transforms into a tertiary follicle. Now a mature tertiary follicle, this tertiary follicle grows in size, and the primary oocyte within that tertiary follicle completes its meiotic division. So meiotic division started when the within the primary oocyte stage itself, that is at the time of when the baby is born, there will be number of primary oocyte, but there the prophase 1 only will be completed, and prophase 1, after prophase 1, metaphase 1, anaphase 1 and telophase 1 will be completed within the tertiary follicle, and the primary oocyte now divides, resulting in the formation of 2 cells. And this division is unequal, resulting in the formation of a large secondary oocyte and a small polar body. It's clear, right? So the primary oocyte completes its meiotic division where? Within the tertiary follicle. And the division is unequal, resulting in the formation of a large secondary oocyte and a small polar body. Now, the tertiary follicle. Now, inside the tertiary follicle, once the primary oocyte completes its meiotic division, what is found inside is the large secondary oocyte. Now, the tertiary follicle further develops into a mature Graafian follicle. So, what is found inside this mature Graafian follicle? It will be the secondary oocyte, which is haploid. Whereas in tertiary follicle, what do you find? The primary oocyte, which is undergoing or completing meiotic division. Now, primary oocyte is diploid in nature, but a secondary oocyte is haploid in nature. So inside a Graafian follicle, we find a secondary oocyte and a small polar body. This polar body later degenerates. Nothing much is known about what happens to this polar body. But the secondary oocyte now will be inside the mature Graafian follicle. Now, this mature Graafian follicle now ruptures, forming Okay, so this Graphian secondary oocyte now after formation will be now surrounded by a new membrane which is called the zona pellucida. So already the primary oocyte will be having a covering, that is a plasma membrane covering. It will be, it encloses a cytoplasm inside. The plasma membrane of the primary oocyte as well as the secondary oocyte is called as the oolemma. It encloses a cytoplasm inside which is called as a ooplasm. Now the secondary oocyte will be forming a new membrane around it which is called as the zona pellucida. Hope it's clear. So now the secondary oocyte, so the first layer, first what we find inside the secondary oocyte is the ooplasm which contains a nucleus surrounded by oolemma. Around oolemma there will be one more membrane which is called as a vitelline membrane. Around the vitelline membrane we find this one, that is the zona pellucida. Okay, we'll be studying about the structure of the ovum later. So for the time being, you should know this much, that is secondary oocyte will be surrounded by a layer, a membrane called as the zona pellucida. Now, the Graphian follicle later ruptures, it breaks open to release the secondary oocyte. And the secondary oocyte now we call as ovum. Actually, it is not the functional ovum, it is a secondary oocyte. Inside the secondary oocyte, the meiosis 2 has started, but it has not completed. It got arrested at metaphase 2 stage. Okay, and the process of release of the secondary oocyte from the Graphian follicle is called ovulation. So what is ovulation? Ovulation is the process of release of a secondary oocyte from the Graphian follicle by the rupture of a Graphian follicle. That is called as ovulation. Hope this much is clear. So at least this much. Now, this whole process of transformation of a primary follicle into tertiary follicle, primary follicle into secondary follicle, and then into tertiary follicle and to a mature Graphian follicle, this takes place. Okay, this takes place within the ovary, and only one follicle gets transformed into a mature Graphian follicle in a month. That means only one ovum will be released out in a month, or per month one ovum will be released out. That means if this month, if it is from the right ovary, the next month it will be from the left ovary. So only one ovum will be released out per month. So how many follicles will be? There will be number of primary follicles, but only one follicle transforms into secondary, tertiary and mature Graphian follicle. Hope this is clear. And the process of release of the secondary oocyte from the mature Graphian follicle is called as ovulation. Hope the whole process of oogenesis is clear to you all. Okay, right. Now, here we have a picture to show the summary of the oogenesis. What I explained only in a picture form, that's it. So, this is the diploid oogonium, which will be found in the fetal ovary. That is why it is written here before birth. And this oogonium undergoes number of mitotic division. Millions of oogonia will be formed, and inside the oogonia what happens is the prophase 1 will be almost completed and it gets arrested, and now we call such oogonia as a primary oocyte. Okay, now the primary oocyte completes. So at puberty, when a child, when the female baby attains puberty, what happens is the meiosis 1 will be completed and the meiosis 2 just begins. As a result of completion of meiosis 1, what happens is there will be the formation of two cells, one large cell which is called as a secondary oocyte and a small cell which is called as a first polar body. This polar body never becomes an egg. The polar body, what happens to polar body is not known much, but it is believed that it degenerates. Now this secondary oocyte now undergoes or completes meiosis 2. Okay, so in secondary oocyte stage, the secondary oocyte stage, meiosis 2 begins and it gets arrested at metaphase of meiosis 2. So during the time of ovulation, the secondary oocyte will be released out. Okay, now, so this is a secondary oocyte. Each month after puberty, one secondary oocyte will be released out. Now at that time, exactly, if there is the entry of the sperm, or if the sperm reaches the fallopian tube, there are chances of fertilization. The sperm enters the secondary oocyte. The moment the sperm enters the secondary oocyte, what happens is the secondary oocyte completes its meiosis too, resulting in the formation of again a second polar body and a functional ovum. So inside the functional ovum already the sperm nucleus will be present. So that is why we call that as a fertilized egg. So the sperm nucleus and the egg nucleus, that is the nucleus within the ovum, fuse together to form a diploid zygote. Okay, so the meiotic division, meiosis 2, will be completed only after fertilization. In case if fertilization does not take place, what happens? The secondary oocyte degenerates. Okay, it will not be, meiosis 2 will not be completed and the secondary oocyte degenerate. Now it is believed that once the secondary oocyte is released out, it has a lifespan of only 4 to 12 hours. Within 4 to 12 hours, if fertilization does not take place, meiotic division, meiosis 2 will not be completed and the secondary oocyte degenerates. Hope it's clear? Alright. So that is about the process of oogenesis. Hope it's clear to you all. Now, let's see what are the differences between spermatogenesis and oogenesis. Okay, so the division is almost the same. So in both spermatogenesis and oogenesis, we find three different phases. First phase is multiplication phase. That is, here in spermatogenesis, spermatogonium undergoes repeated mitotic division. Here, oogonium undergoes repeated mitotic division. In growth phase, what happens is the primary spermatocytes in spermatogenesis, the primary spermatocytes undergoes division resulting in the formation of secondary spermatocyte. Here, the primary oocyte undergoes division, first meiotic division resulting in the formation of first polar body and a secondary oocyte. And in differentiation phase, the secondary spermatocytes again divides resulting in the formation of spermatids. Here, secondary oocyte divides to form ootids, which later transform into a functional ovum. Here, this transforms into a functional sperm. Okay, now let's see the differences one by one. Please note down the first point of difference. Spermatogenesis. Let us define what is spermatogenesis. Genesis means synthesis of sperms within the seminiferous tubules of the testis. That is called as spermatogenesis. What is oogenesis? Synthesis of ovum within the follicles of the ovary. Hope it's clear. First point. Second point. Spermatogenesis begins only at puberty, whereas oogenesis begins during embryonic development. Hope it's clear. Right, that's the second point. Third point. The primary spermatocyte undergoes mitotic division. Right, meiosis 1. Primary spermatocyte undergoes meiosis 1, resulting in the formation of two equal-sized secondary spermatocytes, whereas primary oocytes undergo first meiotic division, which is unequal, resulting in the formation of a large secondary oocyte and a small polar body. Okay, then third point of difference. Secondary spermatocyte undergoes meiosis 2, resulting in the formation of 4 spermatids, whereas secondary oocyte undergoes second meiotic division, resulting in the formation of a large ootid and a small polar body. Okay, hope you got that difference. Now, as next point of difference, you can write, from one primary spermatocyte, 4 sperms will be formed, whereas from one primary oocyte, only one functional ovum will be formed. From one primary spermatocyte, 4 functional sperms will be formed, whereas from one primary oocyte, only one ovum will be formed or produced. Okay, then another point of difference you can see, right? Polar bodies are not formed during spermatogenesis, whereas polar bodies are produced during oogenesis. So these are the different differences that you can write for spermatogenesis and oogenesis. Hope it's clear. Once again, spermatogenesis takes place within the it is a synthesis of sperms within the seminiferous tubules of the testis. Oogenesis is the synthesis of ovum within the follicles of the ovary. right? Second point of difference, spermatogenesis begins at puberty, oogenesis begins during embryonic development itself. Third point of difference, primary spermatocyte undergoes meiosis 1 resulting in the formation of two equal sized secondary spermatocyte, whereas primary oocyte undergoes meiosis 1 resulting in the formation of, which is unequal, resulting in the formation of one large secondary oocyte and a small polar body. Then, as third point of difference, you can write from one primary spermatocyte, four functional sperms will be formed, whereas from one primary oocyte, only one functional ovum will be formed. Then, as next point of difference, you can write polar bodies are not produced during spermatogenesis, whereas polar bodies are produced during oogenesis. Hope it's clear. So these are the points of difference that you have to remember when you write, when you study spermatogenesis and oogenesis. Okay, please study this diagram as the diagram for schematic representation of spermatogenesis means you draw this diagram, and schematic representation of oogenesis means you draw this diagram. Okay, so please see this, and in oogenesis, when you draw, you have to mention that in the previous schematic representation, as it is shown at puberty, at fetal, in the fetal ovary, what happens: birth, childhood, then puberty, then adult reproductive life. All those points also you have to mention. Hope it's clear. Yes or no? Okay. So that is all about oogenesis. So I hope the difference you all have understood clearly the differences between spermatogenesis and oogenesis. Okay, thank you.